Hello friends, today we are going to study about protein evolution. The amino acid sequences are specified by the genes, therefore mutation in genes will alter the protein's primary structure. And once the primary structure get altered, it will affect its 3D structure and eventually it will affect its function. And mostly these mutations are deleterious in nature, but in some cases it can increase the fitness of the host. That means it can be useful or helpful, and that process is known as evolution by natural selection. But mostly the mutations are deleterious in nature. For example, the sickle cell anemia. What happens in sickle cell anemia? The shape of the RBC becomes sickle shaped, and because of this it oxygen carrying capacity decreases. It cannot carry oxygen. And why this sickle cell anemia happens? Because of the glutamate gets converted into valine. And where this conversion happens? In the beta globin chain. In the beta globin chain, when the glutamate gets converted into the valine, this results in sickle cell anemia. And why this happens? Why the glutamate gets converted into the valine? Because of mutation not in the amino acid sequence but in the genes that specify these amino acids. So here glutamic acid is coded by the codon GAG. This codon comes from nucleic acid sequence CTC. So when the mutation happens in this T and this T gets converted into A, now from CTC it became CAC. This CAC will give rise to mRNA not GAG but GUG. This change in triplet codon of mRNA will result in the formation of a valine instead of glutamate. So the mutation in the gene finally affects the function of the protein because the mutation of the gene altered the primary structure of the protein, that is amino acid sequence, and this altered amino acid sequence resulted in altered amino acid formation, which finally results in sickle cell anemia. Now the sequence of the protein reveal evolutionary relationships. That means by comparing the sequences of different proteins, we can trace their evolutionary relationship. For example, this particular protein, that is cytochrome C. Cytochrome C from different species are a single polypeptides of 104 to 112 residues of amino acids. So this cytochrome C is found in all the species and it is a single polypeptide consisting of 104 to 112 residues. Now among these many residues, there are 38 positions where the amino acid is conserved. That is, the same amino acid is present in these 38 positions in cytochrome C of all the species. So these 38 positions are conserved or are highly conserved. There are positions, there are little variations are allowed, but the variation should be chemically similar amino acid. That much variation is allowed. This is known as conservatively substituted. There are positions where different residues are present. So in a stretch of 104 to 112 residues of cytochrome, there are positions where the amino acids are highly conserved. Then there are positions where amino acids are conservatively substituted. That means chemically similar amino acids can replace them. And there are positions where very different amino acids are found. So the protein sequence is very important, and with the help of these protein sequence we can find the evolutionary relationship. Now related species have evolved from a common ancestor, so it follows the gene specifying each of their protein must likewise have evolved from the corresponding gene in that ancestor. Which means if the proteins are related, that means the mutation or changes in their amino acids will be less. If the proteins are non-related, then the changes or mutations in their amino acid will be more. They will be more diverse from each other. So by seeing whether they are diverse or they are related, the sequence of amino acid, we can trace their evolutionary relationship. Now, sequence comparisons provide information on protein structure and function. In this, Few terms we should know. One is invariant residue. Invariant residues means that is same amino acid. Conservatively substituted means similar amino acids. Hypervariable means variable amino acids. So once we can compare the sequence of the protein, from that sequence only we will come to know the protein structure and the protein function. If we have two proteins, protein A and protein B, and protein B is unknown, and after comparing, if we found that these two proteins are highly similar, and suppose we know the function of protein A, so from here we can deduce the function of protein B. So this how we can know the structure and the function of any unknown protein by comparing them with known protein. Now one question is, why essential proteins are also not identical in all species? Correct. That means here in cytochrome C, this is a very important protein. So it should be fully conserved, but it is not like that. 38 positions are fully conserved. Other positions can have similar amino acids, and there are 8 positions where variable amino acids can be found. So why it is so? If it is that important, it should be fully conserved. Then why any protein or any essential proteins are also not fully conserved? So that means why it is so? Because mutational process always continues. But in the important species, these mutation will continue to change in such a way that it should not affect its function. That means mutation is a normal process of evolution. Every gene will go on mutating, but the essential proteins will get mutated in such a way that it should not affect the function of that protein. And this phenomenon is known as neutral drift. Drift means shifting is happening, but that shifting is not affecting the function of the protein. So that shifting is neutral in nature. Though mutation is continuously happening, though shifting is continuously happening, but still it is not affecting the function of the protein. So that process or phenomenon is known as neutral drift. Next, protein evolved by the duplication of genes or gene segment. Now, evolutionary conserved sequences are known as domains, which are about 40 to 200 residues long. The domains whose sequences are more than 40% identical will have the same function or similar function. The domains with less than 25% of sequence identity will perform different function. Now, what are orthologous? Orthologous means these are homologous protein with same function in different species. For example, cytochrome C found in different species. So they will constitute a group of homologous protein which are called orthologous. Now also there are homologous proteins which are found in same species. Now this difference, this homologous protein, they arise due to gene duplication followed by divergence. Means one gene is present, that gene will get duplicated, but here due to mutation this protein will function different function, this protein will function different function. So within a species the similar proteins arise through gene duplication followed by divergence. Now such proteins are known as paralogous. That is, after duplication, when two genes start performing different function, that two set of genes are grouped under paralogous. Example is hemoglobin and myoglobin. Now there are some genes which are known as pseudogenes. Pseudogenes means these genes do not express. So genes has only a limited time to evolve a new functionality before it is inactivated through mutation. And when it becomes inactivated due to mutation, that gene is known as pseudogenes. That means once that gene was performing some function, but now that genes no longer express them. So that set of genes are known as pseudogenes. Now next is the rate of sequence divergence. So this graph, what this graph is saying to us, this is million of years since divergence, and this is number of amino acid that changes during these years. So what we can see here, histone, very less changes over a period of time. cytochrome C changed more compared to the histone, but very less compared to hemoglobin and fibrinopeptides. Next is hemoglobin. Over the period of time, the amino acid change in hemoglobin and more change is observed in fibrinopeptides. So these changes or these mutations or this variations in amino acid is not same in all the proteins. They change over a period of time at different rate in different protein. Now what are the factors on which these rates depend? One is protein function and second is protein structural stability. What function that protein perform, how much important that function is, what will happen if that function gets stopped in the body. So everything determines that at what rate the protein will change or the mutation will change. And second, what is the stability of that protein structure, how much stable that structure is. So these are the two factors that on which the rate of sequence divergence is dependent. So this is about protein evolution. What we learned that protein evolve continuously or the mutation is a continuous process and is a part of evolution. Every protein or the amino acid sequence or the gene will go on mutated, but its rate vary from protein to protein and which depends upon the importance of the protein function as well as the protein structural stability. Doesn't matter how much important the protein is, the full protein or all the amino acid of the protein are not conserved. There are positions where the amino acids are highly conserved. There are positions where amino acids are conservatively substituted and there are positions where variations are allowed. In case of important proteins, whatever mutation happens, it happens in such a way that it should not affect the function of the protein, which is known as neutral drift. So this is about protein evolution. Now one question from my end for you all. Below is a list of first 10 residues of the beta helix in myoglobin from different organisms. You have to tell me which positions appear unable to tolerate substitution, which position can tolerate conservative substitution, and which positions are highly variable. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.